Welcome to Cracking Cancer, where we tackle the tough questions about cancer with real patients and world experts. Our mission is to empower the cancer community through education, bring transparency to the oncology system, and drive funding for critical cancer research. Whether you're facing a diagnosis, supporting a loved one, or simply here to learn, we're here to bring clarity, hope, and action to the fight against cancer. Hey everyone, welcome to Cracking Cancer. I am your host, Dr. Kyle Concannon, physician scientist now at the University of Colorado. And I am joined here with Samantha Murrell, partner in crime. Samantha, maybe a word about yourself?
Hey everyone, Samantha Murrell. I'm a patient advocate and I have stage 4 non-small cell lung cancer with a HER2 mutation.
Well, thanks again for joining us, Samantha. And today we're talking about cancer research. I think there's a lot of misconceptions and nobody really knows what that term means. So hopefully today we can demystify the term a little bit, dive into some of what cancer research is, some of the myths, what it's not, and educate people as to what conceptions may be out there and what the reality is behind that term. So Samantha, I think you may start us off with some questions that you have as someone who isn't integral in the cancer research field. And for those listening, my role as a physician scientist is I'm predominantly in the laboratory. So most of the time I'm doing cancer research, and less than half of my time I'm actually seeing patients, but I do still see patients in the oncology clinic. But as a physician scientist, a lot of my time is focused on the cancer research part. So hopefully I have some insights to add and we can demystify some things, but I'll say it's a big, complex world and not even I understand all of it. So I'll answer what I can and hopefully we can, get some insight there and take it from there.
Yeah, I'm really excited about this topic and debunking some myths potentially as well while we go about it. I guess let's start with the absolute basics because before I was diagnosed with cancer, I didn't know there were so many players in this space. I didn't know people like yourself existed. I thought it was all drug companies. So I have so many questions. But basics are like, who are the major players in the space of cancer research?
Yeah, it's a big question. I would say the major players range from academic institutions like the hospitals that I've trained at, the University of Washington, MD Anderson Cancer Center, those places that people think of as, treating hospitals. But then you also have preclinical institutions that do the research. So these may be institutions that don't see patients, but are doing the biology, the investigative biology, and learning more mechanism stuff. But then you also have government entities that are funding the research and they're kind of determining where things are going to proceed and what gets funded and what doesn't. You have independent advocacy groups that have a shared mission and they're funding and helping to shape the route of cancer research. So there's a lot of integral players. The challenge, I would say, is that not all the integral players work together on everything that they could. And, behind a lot of this is pharmaceutical companies too, that are probably I would say the largest champion of cancer research is coming out of pharmaceutical companies. Yeah. So I'm curious to hear now your perception when you had heard cancer research. What did you think cancer research was coming from, or what did it entail?
I mainly thought cancer research was coming from drug companies and then government entities having some method of funding towards the research, but it was mainly being done by drug companies. So knowing that there's a lot of other players involved is super interesting, but super frustrating that they don't necessarily all work together cohesively. And my initial reaction to you, like listing some of these key players that I didn't know about, is how does one decide what you're going to be researching?
Oh, geez. Yeah, that's a tough one. I think part of it is, if you back up and you just take the individual who's going through the process and they say, I want to do cancer research. So that individual, when they're earlier in that process, undergrad, grad school, typically they have some sort of academic interest, which is I would say for most people, the primary driver. It's something they find interesting and they want to learn more about that. And they think it sounds promising and meaningful. And then that probably is the nidus of where the cancer research bug is caught. And then after that, you start with that and then you walk down a road, toward that direction. So let's say for me, for example, it's like lung cancer was interesting. I thought learning more about targeted therapies was interesting. These are the therapies that we've learned can specifically attack the distinct cause that a cancer occurred. So for you, you have a HER2 mutation. I thought it was so interesting that we learned about these mutations and we found very unique ways to target them, like a sniper or, something very, very specific rather than the nuclear bombs that we were doing, which is chemo. So I was very interested by this idea, this topic. So I went down this path and I found opportunities and I learned and I grew in that regard. But later, so that framed everything. But later, your opportunities are around largely funding sources. You have to do what is going to get funded. And I think on one hand, that's kind of a good balance to make sure that people are investigating things that the greater community deems worthy of investigation, worthy of tax dollars, worthy of the hope and work that patient advocacy groups put in to raise funding. And on the other hand, that tends to lean more toward things that are directly translatable, meaning you research something that can go into clinic. And there's a clear line of sight from where you start and where you're going to end. However, if you think about it, most of the Nobel Prizes, the major advancements, occur from a scientific interest that you didn't know where it was going to go. So we do lose a little bit of the basic science. We'll probably talk more about what these different terms mean, but that like understanding how receptors work and understanding the nitty gritty biochemistry, that doesn't clearly translate into clinical change. We lose a little bit of that at the cost of, or I should say at the benefit of, something that's more directly applicable to patients. So that ends up directing us toward where our careers go. You have to follow the funding sources. So I know I've talked to you recently about, a grant that I'm applying for that, has to do with her too. And, we have to think about what are the people who are sponsoring this grant interested in championing and what do we think is the most promising thing within that realm. I can't come up with an idea that I think is intellectually interesting. And who knows, maybe that'll be a grand slam and 20 years from now, that'll lead to a Nobel Prize. The likelihood of that's pretty small. So we have to do the stuff that's believable, fundable, interesting to the funding source, and sometimes something a little more safe, because you want to champion the hope that people have when they work hard to raise the funding around this.
So let's get back to the basics in terms of like, let's talk about funding, right? Because you're using that term a lot and saying that it dictates in a lot of cases what research is done. So what are the different avenues that funding comes from?
For most people, they're getting funding from a large entity like the government. So those are probably the biggest sources of clinical funding and laboratory funding for someone who's not in pharmaceuticals or, for someone in an academic center like myself, probably the biggest source is the government. After the government comes, I would say, private donation groups like, I don't want to, isolate specific ones, but enter whatever research organization. There's usually things that'll rally around a lung cancer or prostate cancer or whatever. They do their 5K, they raise their money. And then what they actually do is they set up information on their webpage and they put out things to researchers to say, hey, we want to fund something in the breast cancer space targeting, in this case, maybe still HER2, and, give us your best idea on how to do that within these parameters. And we'll give you 250,000 over 2 years. So that would be another example of something that's very, very common. Beyond that, you can have partnerships with pharma. Those can be Those can be very helpful because the pharmaceutical company, maybe they make a drug and they want to use someone else's models that they don't have to prove that the drug works and to investigate it further. So they'll pay a lab to work together. And then the lab publishes the paper together. And that's usually actually a pretty good example of pharmaceutical companies working well with academics.
Mm.
I would say those are probably the major ones. But again, the biggest one is going to be the government. And this is what you talk about, or you hear with NIH funding. Department of Defense actually pays quite a bit. So through different avenues, the government pays for a lot of the academic cancer research funding. And that's why a
So,
lot of us feel threatened when there's variability in how the government does that.
and you read my mind. So I was going to say quite a potentially controversial topic, and we won't go into the politics of it, but there's been a lot of attention on dramatic cuts at the NIH. And, I read a lot about the potential devastating consequences that could have on cancer research. And, I go back and forth in my head, like, is it really that bad? And then, I hear from you who says, like, yeah, the majority of funding comes from the government. And
Yeah.
so, like, how devastating are these cuts?
I would say the full consequences are to be determined.
Yeah.
And, because we don't actually know yet everything that's going to shake out. So right now, I would say there is damage in the speculation. People don't know how to plan their laboratories. They are submitting these large grants to the government, and there have been delays.
Hmm.
And what that may, just to frame it, what that might mean for a researcher is they have a multi-million-dollar grant over several years. And if there's a pause in that, then that funding isn't distributed and someone's payroll doesn't get made. So there are layoffs. Right now, as I'm setting up my laboratory, I have been interviewing many, many people who got laid off from just really the ambiguity around it's not clear that I'm going to be able to fund you.
Hmm.
So that does cause disturbance. And when that trickles down into trials and things like that, which also are frequently funded from government sources, then yeah, there's a very strong ripple effect.
Yeah.
So, yeah, I would say there has been a real consequence. What it's ultimately going to settle down into, who knows? Maybe there will end up being more funding for cancer research in the long run. That's my hope. But in the short term, anything that prevents you from being able to make a long-term plan is disruptive. And there's no way around the fact that the ambiguity right now is tough. And I mean, I'll say my hiring almost got paused uh, because if they don't know that the NIH is going to keep renewing funding, they can't hire people with these research funds earmarked. So what tends to happen is when there's ambiguity, everyone locks down, holds on to the money they have because they need a safety net. They need to keep the people that they currently have employed to the best of their ability. And yeah, that really stagnates progress. And then the high risk, high reward things that lead to the new innovations are not going to be attempted, or at least not to the same degree. So
Yeah.
you're going to do the safe things that just keep you moving, and you want to secure your funding and kind of hold on to it.
And I think this is a great segue to talk about, when we talk about funding and funding a lab, like I default to you're literally paying for the samples and the testing and everything, but it's a lot more than that, right? I mean, there you have staff that I didn't even think about were covered under the grant, and other things. What are all the things that are entailed into the cost of doing research?
I think you nailed it. The staff are probably the biggest one.
Yeah.
And so the staff is, well, it's the biggest not only from a monetary standpoint, but at least for me as like a new physician scientist this is my first time being in that leadership position, and your decisions impact someone else's like their life for real. They are going to have the financial stability they need to take care of their kids, or they're not, based on how you do. And so I would say that's the biggest burden financially, psychosocially, all the things is now you're a leader and you have responsibilities over other people's outcomes. And that's a big one for all the reasons. After that, you'd be surprised. I think when a lot of people think of a cancer research lab, they're thinking of patient specimens, they're thinking of, the blood samples. But I would say a lot of the labs I know of, we're doing stuff that the public isn't fully aware of yet. And that's one thing that I think we really want to change with this podcast platform and then some of the stuff we're going to do in the lab is really detail what that is. So, for example, let's say I get your patient tumor. So I'm going to give you an example. And you got treated with these therapies, and I want to know what would be the next best thing to do when resistance to your current thing occurs. And I'll say, well, I'm in lung cancer, we'll say EGFR. That's a really common mutation. It stands for epidermal growth factor receptor. A lot of patients with lung cancer have the EGFR mutation. But what do you do once that stops working to the best therapy, inhibitors of EGFR? So what we could do is we could take a biopsy and we then bring it into the laboratory in tissue culture hoods, which one had to buy. And then in that are the petri dishes that you may have seen, like the nerdy scientists always have their goggles on. And sorry, you're
No, you said something about tissue hoods and that you had to buy, and I have no idea what you mean by that, and I'm sure many people won't either. So can you
so
explain what that is?
so when you're working with patient samples like a tissue culture, so let's say we got a patient's tumor and it's in the
Yeah.
hood. I keep using the hood now. So it's in culture. Like we want to grow it so we can study it. Well, that has to be sterile. Like you can't just be in the counter because stuff's going to fall on it. It's going to get infected with stuff. So we actually have sterile spaces that we have to maintain.
Okay.
And we spray it down with ethanol. We clean it before and after every use. It has a constant HEPA filter that's running. So your hands go in this hood, in this
Okay.
protected environment, and you have to do everything to those cells in the petri dish in that hood. If it's out of that hood, it's always covered. It is
Gotcha.
never open.
Okay.
So you have
Thank you.
to maintain these systems, and then it goes out of the hood into an incubator, which is 5%, 4% CO2 and 37 degrees. It is maintained perfectly. We test it for different infections. Commonly, mycoplasma is the big one we test for. So you have to make sure that everything you're managing is under ideal conditions. So just the maintenance, and that's just one small example of things we do,
Mhm.
but the maintenance of the infrastructure to do research is costly. So that's another big cost. After that, you get the reagents. So these are the things that you need to do to your day-to-day work. Those cancer cells, they're on a tissue culture plate, a petri dish. Well, that petri dish is a sterile petri dish that you opened in that hood, in that sterile environment. Anytime that bag is open, it's always in there. But those cost money. You can't just buy that at, Staples. So you have to buy those. You have to buy the media, the solution that the cells are growing in. And that's a specially made sterile media with sterile fetal bovine serum. Instead of your own serum from your blood to feed cells, we use fetal bovine serum, calf serum. So we use, cow blood essentially to, so that's what that is. It's they took all the red blood cells out of cow blood and they just use all the good parts, the nutrients from cow blood. And we add that to be a nutrient source for the cells. And all these things cost money. And then you haven't even done an experiment yet. This is just to like have it. So then
I
you
have so many questions because bovine colostrum is in my protein powder. Like,
oh, I mean, yeah. The poor cows, they're doing a lot of heavy lifting here. But yeah, it's a good source for a lot of the nutrients that cells need. In your blood is all the nutrients your cells need.
right.
Cow's not that different. So
Yep.
um, so yeah, and then you have to pay for if you want to do experiments with drug compounds to see if they work or don't, you have to buy them. Then you have to get different plates if you want to do different experiments. You have to read your experiments in some way, interpret your data. So that might require a plate reader. Like there's just so many things. And the amount of things that you need is, changes by how much, by what you want to do. So if you want to do really cool, fancy science, then you're going to need to pay more money and get fancier equipment.
Mm.
Um, and that's not even including the stuff that if you want to do the DNA sequencing like you got on your tumor, we do that a lot in the laboratory. You might send that out to a company or something to do. And then you have to pay sometimes people to analyze your data with you.
Mhm.
Like I'm not someone who will sit down and look at every mutation that came out of your entire genome and know how to make sense of that right away. So I may need someone who can sort the data and interpret the data correctly to work with me, and you have to pay them too. So there's a million fees in the laboratory from personnel, reagents, collaborators, and that's not even getting into what happens if something's promising and you want to push it forward into trial. There's so many additional costs that occur, and it's interesting because we talk about costs. That's I think everyone knows that's the biggest barrier to cancer research progressing forward is the cost to it. And a lot of times that's how a good idea ends. Is you have something that's a good idea, it looks really promising, but you just don't have the money to pursue it. To pursue it appropriately would cost a lot, and you have other things that are safer that, are not as costly and they'll get the publication out, and that's what you're going to prioritize. So the cost ends up being a primary driver in what we do, how we do it, for better or worse. But more money would definitely be better, in my opinion. So anything we could do to advocate for ideas for more cancer research funding, I'm on board with.
Yeah, I think that's the frustrating thing from my standpoint. The more I've been learning is there's so much research or similar research that's being done over and over and over again, or like just a little bit of a tweak or a little bit of a change. And it's like, is that really how we're going to find out like this incredible, amazing thing if we're just sticking to the same things? And it's frustrating from a member of the public's perspective of like, as an example, T cell therapy, right? That was, I'm sure, kind of out there at the beginning, and it super excites me. So, like, how something like T cell therapy, how did that suddenly come about when it's kind of more out there than the some of the other things? Because to me, I'm just seeing similar.
Yeah.
And T Cell was like, ooh, that's super interesting and cool. And I'm just curious how we can get more, ooh, that's super interesting and cool things to be done in research.
Well, let me answer your question with kind of a continuation of that thought. So, the challenge with a lot of cancer research is you invest so much money into something to see if it works. And it's really hard to know how much money to put into an idea that sounds good. And when do you put that money into something different? Because there's opportunity cost everywhere, and the example with T cell therapy is T cells are incredibly expensive. This is a very, very, very expensive tool. And I don't mean in the laboratory. I've done a lot of experiments with T cells in one way or another. But if you're going to do that for patients, those are very expensive studies. So the level of information that you're going to need now is higher to prove that this is going to be worth it and promising largely for a company to take it and be willing to process it with their donors and such. So it's tough. The promising stuff, you want something that's out there, novel, new, but it has to be promising enough to warrant the amount of money that's going to need. And that's really hard to balance. You need really, really good signal. T cells have been tough as your example because they sound really promising. Occasionally they are, but in solid tumors, T cell therapies have not been very good, and they're very expensive. And you don't know, like, we found some good signal. And is the difference between, let's say, 10% benefit and 90% benefit one thing we haven't found yet? Like, you don't know. You really don't know. Are we right at the precipice of the break point where we figured it out? And that's the challenge with research, is it's always risk. And it's tough to be a donor, or let's say you're the person who's determining which grant to fund. You can say, okay, T cells haven't really been that great in solid tumors for the past 10 years. Are you the person out of the last 500 that did find the difference maker? Or are we going to try this other thing that is an incremental gain? Maybe we'll make a current therapy work 10% better. Many people will take that.
Yeah.
They'll say,
Yeah.
I'm going to take the incremental change over the likely nothing.
Right.
And the reason I think that is just because we don't have enough funding.
Yeah.
If
And
you
I see
have,
that, I see that often in some drugs, right? There'll be a new drug for a new mutation or just a cancer that doesn't have a mutation. And they'll be like, well, the progression-free survival is a month better.
yeah,
And it's like, woohoo!
yeah, you're celebrating this? Yeah. Yeah.
Wow. Like, and I don't want to poo-poo it too much. Like, another month is great, but like, it's those baby steps of like, come on, like, can we do a little bit better here? And I guess you're saying where the funding sources are coming from, and they're kind of dictating what is being done. But like, as a researcher, if you do think you have solved this amazing problem that no one else really knows about or understands yet, how do you garner attention to it to get research, or is that not even a thing that happens?
So I think it can be a thing that happens, certainly. We do get breakthroughs. And a lot of times you have to come up with the idea and fund it on your own somehow. So a lot of laboratories do have some discretionary funding, which is like, this is a pot of money that is earmarked for nothing in particular. And then you do get to do, your whatever idea. Now, if the idea is not too costly, there are small grants called, pilot grants. And that's their goal, actually, is that it's $50,000 and they say, hey, give something bold a shot. And so that niche is addressed in some way. There's just not that many of them, and you can imagine it's really hard to evaluate crazy ideas. Like, one example, one of the most effective therapies that's ever been generated in cancer is the bone marrow transplant. So for those who don't know, a bone marrow transplant is when the oncology team gives super high dose chemotherapy, obliterates all the bone marrow. So this is the cells that make your red blood cells, your platelets, your white blood cells that fight infections. All those cells die off. But before you did that, you took some of those things from the patient. Once they all die off, you kind of re-inject them to regraft all of the bone marrow. So the idea is that you kind of kill off all the cancer, and then, in theory, whoever is giving the marrow back, sometimes it's yourself, sometimes it's other people, but that can restore a normal state. This can be useful in leukemias and things like that. So that is a crazy idea. Like, that is an insane idea. Mm-hmm. And the mortality rate when they first started this was not zero. I have a brother-in-law who underwent a bone marrow transplant and nearly died. Many people nearly die, and many people do die from a bone marrow transplant. So that was an insane idea, and it won a Nobel Prize. It was effective, it worked, and rightfully so. But someone had to say, yeah, we're going to do that, and come up with this crazy idea, and someone had to fund it, and the patients are in the hospital, which is very expensive. They're monitored, they're totally immunosuppressed. So someone did make that leap, and it was totally worth it. But, for each of those, there's 5 examples of someone who did a really bold thing and it didn't work out.
Yeah.
And if you're the person who's determining who gets funding and who doesn't, which crazy idea is just crazy enough to work is a tough call. Like,
Yeah.
you're just not going to get it right. It's complete gambling. That could have just as easily just killed everybody. I mean, really.
Yeah. And I think, you and I talk a lot about supplements and other things that people do in addition to their cancer treatments. And, a lot of people online will say, well, why hasn't there been a trial on, I'm just going to randomly pull a vitamin out of my head, turkey tail. Why has there not been like in-depth research on turkey tail? Because in Japan they give it to people as part of their treatment, just as the norm. And so people say, why hasn't X had a trial or whatever? And it makes more sense to me now. It's because if you're trying to look for funding and do
Yeah.
research, it's going to be really hard to be like, hey, I want to do a trial with mice on with turkey tail, and someone fund me, government, and like, what are the long term benefits of that, and what can you prove? And so I think there's a lot of conspiracy theories around like, why are people not researching these supplements where there's technically no money to be made? And they kind of say, well, like, it's not being done because no one's going to make money if we find out that it's successful, but it's really because it's freaking hard to get funding for this stuff, so people just can't do it.
Yeah, I would say, I mean, I have a lot to say on that. Um, I think people don't understand always just how expensive trials are.
Yep.
Every patient enrolled in the trial is thousands and thousands of dollars. And the number of patients you need to enroll to see if something works or not depends on how strong you think the effect is.
Hmm.
So, an example, if we thought turkey tail was 100% curative, you took it and it killed cancer always,
Hmm.
we would not need a lot of people for that trial. You
Right.
could get 15 people on one arm, they get the turkey tail, 15 people don't, and you will see immediately that that is true.
Right.
But if the benefit were, like you were saying, a month, you need 100, 150 people in each arm, depending on how aggressive that cancer is, to
Hmm.
determine if there's going to be a difference.
Right.
So, and then you have to ask, okay, do we think turkey tail is so beneficial that we're willing to make a chance on this 15 person trial? But if it's not that beneficial and there really is a difference, it's just not that big, you missed it.
Hmm.
No,
Right.
and that was a total waste, wash.
Yep. Yep.
And then you have to do the big trial anyway, which is going to cost millions and millions of dollars. And to your point, someone has to fund it.
Yep.
And if a pharmaceutical company does have something to gain, that trial is more likely to be funded. That is
Right.
true, for sure.
Yep.
But here's the flip side. Pharmaceutical companies really want to make money. If they thought turkey tail worked, guess what? There's going to be a pill for that. They're gonna sell it.
Right.
So I
Right.
think you can take that purely monetized view and extrapolate that to say, it probably doesn't work because the most profit driven groups on the planet aren't interested. Uh, and you know they'd have their own formulation of turkey tail whatever compound that they would patent and they would charge $1 million a dose. Um, so I think there's a lot of considerations there. Now, those questions can still be evaluated. So what you're talking about is what we would call prospective. So meaning we enroll everyone, we start the therapy, and then we look very closely to see what happens. And that's the best kind of trial you can do. That's the best kind of experiment. Is everyone kind of starts on an even plane. We randomly give one person one thing, and another person doesn't get it from the same random chance. But that's not the only way you can evaluate something. We can look retrospectively, meaning let's look through our records. These 50 people were taking turkey tail, these 50 people weren't. We don't tend to stop people from taking something unless we believe it to be harmful. And turkey tail usually doesn't fall in that category. So we can look in the past and say, did those people do better? And I promise you the cancer community would be interested. Like we recommend acupuncture for certain things. Like we don't, no company is getting rich off of the advent of this thousands-year old,
Right.
like no one has patented acupuncture to my knowledge. So yeah, I mean, we are interested in those things. People do look at those things. The challenge is just a lot of the things that you hear about in the community, there wasn't a lot of signal for.
Yeah.
And it's tough when we don't think something's going to be promising. Like it's not promising enough for us to spend our time on it to convince people of that.
Yeah.
So I think that's
No,
really hard.
yeah, it's a super interesting topic, and I think you've shed a lot of light on some of the items that people think about. It seems to be a top of mind for many people out there of like, why isn't X supplement being investigated? And like, it's just not that simple.
Yeah.
There's a lot to it.
Yeah.
But I was, yeah,
So,
go
so
ahead.
one thing that so let me, as a cancer researcher, for a lot of your career, you're trying to be impressive enough to keep doing what you're doing because there's not enough funding for everyone. So you have to keep doing the thing that is going to be publishable, that you think is going to work. So those high risk things like let me run a full trial, get all the money I have and do a turkey tail trial that I don't think is going to be promising, like no one's going to bite on that. And then you reach this phase in your career for some people where they do have enough money to do some of the high risk things. And I think the perception changes. Then it becomes, and you've learned a lot. So you have a big fund of knowledge. So then assume the next thing you're going to do works as well as you think it could. So to your example of T cells, I could spend $1 million on a trial comparing turkey tail to not having turkey tail in patients on chemotherapy who have lung cancer or something. Or I can do the next incremental benefit of T cells. And let's see if maybe we're not going to get them to 90% working, but 10 to 20. We'll do that next jump. If I'm assuming the next thing I'm going to do is going to work, I'm going to do the T cells because I think the increment of benefit to the community is going to be better than maybe the 2 weeks of added benefit that theoretically one might get with turkey tail because we didn't see signs anywhere else yet. So that's another interesting way to frame it is like assume the thing you're going to work on works to the best of its ability. Now, what do you do? So as a researcher, you now have to choose what do I actually think is the most promising thing to the cancer community? Because I only have so many years on this earth and I want to spend them doing the most good. And these incremental, oh, isn't that interesting? Like, why hasn't anyone done the trial on this supplement? It's just the amount of benefit that most of us perceive to the community is so small, if present at all, that we're trying to do the next big thing.
That makes sense. That really helps. That really does
I
help.
know.
No, it does. It does. And I have many questions coming off of that. So, you talk about increments and doing things on top of existing research to better it. Why does research, and I want us to backtrack in another section of this conversation about doing research on mice versus in humans and when do you take that leap and everything, but we'll put that to the side for the moment. How why does it take so long for research to be done to actually like come to fruition where a paper is actually written and published with information? Because, and I'll use this as an example that I don't know enough about, so it's maybe dangerous for me to mention it too much, but you probably know more than you would do know more than I do on this. But as an example, recently top of mind, EGFR. EGFR people that you mentioned, they take a pill called Tagrisso, usually when they're first diagnosed at stage 4. They found recently that adding chemotherapy in some circumstances can better progression-free survival.
And overall survival.
Right. So why did it take I don't know, 5, 10 years
Yeah,
to
longer.
prove that adding chemo made that much of a difference. Like, that's kind of wild and really frustrating.
So this is going to be a very unsatisfying answer, but the answer is
Okay.
that it's super complicated. I
Okay.
I think that it depends on what you're adding the chemo to. Like you could do this same trial with stuff that isn't that Tagrisso, and I would expect it not to be helpful.
Okay.
The idea of just adding more stuff doesn't always pan out to be better.
Right.
And I think that's the pendulum is swinging now in the community to being a little more aggressive. But speaking to this like iterative nature of everything we're doing, and this is why everything happens so slowly, is a lot of times what happens is we look back on experiments that already happened. We added too much chemo to other stuff and made people sick, and their survival was shorter.
Hmm.
And then you have that framework to say more is not always more. We need to space our stuff out. And I'll say there actually are caveats to that experiment that I'm not going to get into here to that trial, but um, off the knowledge that adding everything together in the biggest quantity of things that you could might be worse, then you have to really be careful about when you try that,
Hmm.
because that's a big trial to get to first line to try this. It's very expensive. And to your point about funding, like that is an already approved drug.
Mm-hmm.
So no one makes more money off that trial. On one
Okay.
arm, you have the Tagrisso. On the other arm, you
Yeah.
still have the Tagrisso.
Right.
So like who's funding that trial? And that's a
Gotcha.
real pragmatic thing. It's not necessarily agreed, from the pharmaceutical companies, or at least I wouldn't think of it that way.
Mm-hmm.
It wasn't intuitive that this would be better.
Gotcha.
And there are still a lot of circumstances where we're not sure it really is.
Yes. Yeah, there are caveats.
So to like I was mentioning earlier, to invest that much money in multiple different institutions, many patients, you have to have independent people interpreting the data, independent people reading the scans. These are multi-million dollar trials. And you really, really have to believe that this is going to work. Um, and it's tough. Like we've done other trials like that and they didn't work.
Yeah.
So uh, I think the difference with this kind of therapy is how well that therapy works. I
Mm-hmm.
think if you tried the same thing with earlier generations of it, probably wouldn't have been as good.
Yeah.
But yeah, you don't know when that threshold is crossed until you do the experiment again. And now we're going back to being more aggressive with surgeries and more aggressive with radiation than we were in the past because the therapies are getting better. So we've got to redo all these trials again, and someone's going to have to pay for that. The flip side is sometimes people just do stuff and it's not a trial, and you just do it anyway,
Yeah.
and that gets into our insurance system, but because you can do whatever you want, it just may not be covered.
That's why I like my new oncologist. He's very much of that mindset and just like, you're young, let's think outside the box and do some potentially crazy things that pharmacist insurance might not agree with, which I greatly appreciate, for me personally. But yeah, like you said, it's either that or you're waiting 5, 10 years for to
Mhm.
trials to prove that something is more effective or not.
Yeah, and it does take that long if you want to look at it and do it right and know
Yeah.
for sure. It's going to take 5 years, and hopefully, like the way the trial shaked out is accurate, because there's always a chance that a bunch of people got sick that day. There was a international pandemic that kind of messed up your results a little bit, for a arbitrary example. Um, so those things can happen, and they cause big delays. And everyone wants to be on the arm of a trial that has the thing, but if we knew it worked, we wouldn't be doing a trial. So a lot of these really do have toxicities. They have side effects that are worse than not having it. And we have to run the trial to figure that out.
Yeah, I won't lie, the phase 1 clinical trials always make me a little nervous because that's when they're trying to figure out the dosage in humans, and it's like you really are a guinea pig.
Yeah, that is absolutely true.
Yeah,
And it's, it's cutting edge. It's the new stuff out, but we don't know if it works.
yeah,
Um,
yeah.
and, and that's not even that's not the goal of a phase one trial. So for those who don't know, phase one trial are the earliest trials. These are the things that are first time in a human being. Phase two is like the next step, and phase three are the big trials that determine if something's going to be standard of care usually. And the goal of a phase one trial is to determine if something's safe. Like, can I give it? We'll start gentle and we'll slowly go up. And okay, did your eyes bleed? No. All right, let's go to the next one, next dose. And then the phase two is like, we figured out a dose we think that's safe and it probably works. So let's do the phase two trial to say, is it effective? Do we see tumor shrinking? Is whatever outcome we want happening? And then you get to the phase three, which is like, okay, we're pretty sure this thing works. It does kill cancer to some degree. Is it better than what we do now? That's the phase three trial. So a lot of people think like, oh, I don't want the placebo. And rarely do we do placebo trials. Occasionally it happens, but most of the time, the goal of a trial is to say, hey, we have this thing, we think it's promising, and we want to see if it's better than what we're currently doing.
Yeah.
And I think what many people don't know about research, every step along the way, from being in the laboratory, understanding how receptors work, to then figuring out, okay, this is this thing that causes cancer. How do we treat it? So we come up with drugs, molecules, and that's usually pharma companies will synthesize these and work with groups to test them. And then, okay, we found something, we think it works. Let's try it in mice. Does it kill the mice? No, the mice are doing okay. Okay, let's try it in humans. Okay, did it kill the human? No, okay. And so and so. Every step along that path mostly fails. Almost everything fails. I think this is what people don't understand about cancer research is there's so much failure. From the things you're looking at to is this going to be a promising way to attack cancer? Most of that isn't. Once you find something that's promising, what are we going to do about it? Most of the stuff you try doesn't work. The stuff that you do try, is it going to work in mice? I would say a handful of those work. But then does that translate into the human? I don't know. And then, is it safe? Is it tolerable? Did it hit other stuff in the human that we didn't know it was going to target? Does it cause you to, be really sick? Does it, damage your liver? And then to make it through all that way to being an actual therapy that we're going to give almost never happens. So you keep seeing all these what we call Me Too drugs. These are, we found an inhibitor of this EGFR protein in lung cancer. Well, we're going to make one that's just like it with like a little change because,
My favorite topic.
yeah, just the tiniest little change. We think that might bind a little bit better, maybe 10% better. But we do know that it's going to, in all likelihood, pass through all those earlier steps and it's not going to make your eyes bleed. And, maybe it'll be a little bit better. But that's like a safer thing to do than to waste the $1 billion in, funding from all those things just to fail at the finish line.
Yeah.
So I think that's why you see all those incremental changes, like you're saying, and it's tough. It's a really harsh world where your value and how you're viewed from everyone externally is how many publications you have, how much stuff have you contributed. Now that is very different from did you make meaningful change? Those are not
Mm.
the same thing. You can have 50 publications and have contributed virtually nothing to the improvement of cancer biology.
Which is a really good segue again because I read a lot of research papers and I also send them to you and you always say like, but this was just in mice. And there is so much out there that people tweet about and get excited about. especially in the social media realm, and it's just in mice. And you have educated me very well in realizing that, well, I mean, I know obviously mice are not humans, but educating me that, like, it's so different.
Yeah, it's very different.
And so, can we talk a little bit about that? And then also, like, when you do see something successful in mice, what makes you transition to the humans?
Yeah, that's a good question. So if we're trying to figure out what are the differences between mice models and human models, I had an old professor that used to say, um, humans can be rats, but rats can't be humans. So to say that they're very, very different model organisms. Usually, when we're talking about mice, we're talking about immunocompromised mice. So these are mice where I could take your tumor, inject it under the skin of a mouse, and grow the tumor. If the mouse had an immune system, it's not going to tolerate your foreign cells in there. Like it'll just kill everything. So most of the time, the mice doesn't. They don't have an immune system. We do have mice with immune systems, and we do run those experiments too. They're a lot more expensive usually, and they're more complicated. But even then, the mouse immune system isn't the same as the human immune system. Like truly, there are differences, like important ones. So most of the time, when we're talking about mice, we're talking about immunocompromised mice, and it's a human's tumor in it. So you can imagine the interplay of the hormones, like the microenvironment of the tumor, the stuff that in your native lung, the tumor that is in the flank of a mouse under its skin, like that stuff's just not around. So there's a lot of variables there that we've just ignored just for the feasibility of being able to do it. So a lot of times what we end up doing is, I'll give you an example. We have a therapy that we think is really interesting. We learned out of the petri dish that I was talking about earlier, if I apply this at a fairly low dose, the cancer cells will stop growing or they'll die. Like, okay, that's promising. Cool. So we do the thing I was talking about. We take those exact same cells, we put them under the flank of a mouse, and then we give the mouse that drug. And what I'm looking for in that kind of experiment is to say, is that drug going to kill that mouse? Like, is this a promising avenue from a toxicity standpoint? I would probably expect the tumor to do the same thing in the mouse that it did in the petri dish. Or at least, it's probably going to react similarly, unless there's an issue of metabolism or absorption or toxicity. And that happens a lot. Like you do give the dose that is effective against the tumor, and the mice stop eating, they lose weight, and they get really sick. And you say, ooh, all right, that is a big red flag. I don't feel comfortable putting this in a human, even though it totally killed the tumor cells. And this is a realm that I think is really important to talk about because a lot of people will see things before the mouse. They see things in the petri dish and they say, this kills cancer. And it's like, anything kills cancer, truly. Like you can take a dose of whatever you want and I will kill cancer cells with it. Um,
I
you know,
read today that plums can kill cancer cells.
if you I don't
If
know
you,
what they were doing, but I think if you smash a plum against a cancer cell hard enough, the cancer cell will die. If you grind a pit and just put it in that media, it will kill the cancer cell. Promise you. Um, so I'm curious to see what that ends up meaning. Um, but yeah, I mean, you can make whatever data you want if you're not being a good steward of science. And this is where the whole idea of peer review comes in, which we didn't talk about. But this idea that other people are looking at what you did and judging whether or not it's reasonable or not. So you'll see sometimes this stuff floating around in the ether. You sent me an article about this that was
I
not in a
did.
peer reviewed journal.
Yeah.
And I was like, this is already red flag number one, two, three, four, five. Because if someone who knows what they're doing wasn't willing to accept this, then there's a big flaw in the methodology. One of many, potentials, but it's probably super flawed. And I wouldn't wait a lot on something that's not in a peer-reviewed journal. So that's one metric to say, is this thing that's floating around that people are saying cures cancer credible? Now, there are actually things that are out there that are in the conspiracy theory world that I think there is some credence to. It's just taken too far. And this gets to how long things take to prove something works. The burden of proof is actually really, really high. because we have so many assumptions that you have to make. So we have to do a lot of experiments to prove that something is worth investigating, and we actually think it's going to be more helpful than harmful. So there's a lot of boxes to tick. It takes a long time, and if you're wrong and you rush it, you end up wasting a ton of money, a ton of hope that that could have been targeted elsewhere. And as stewards of the scientific progression in cancer, like that's our utmost duty, is to make sure that we're good stewards of funds and we're good stewards of hope, and that we use these things appropriately. Because there's opportunity cost to everything you do. I only have so many hours I can work. My team only has so many hours they can work. And if we're working on one thing, we're not working on another thing. So you really have to choose.
So how long does the petri dish and the mice phase usually take?
It depends on how fleshed out your idea is. So if someone hands you a compound that you think is effective, I could tell you in a number of different cell lines, you always want to do more than one because one might just be a fluke. That could have been a uniquely susceptible cell line for whatever reason. It's still interesting, but not generalizable to everybody. So you want to do a couple of cell lines. Um, make sure your dosing is good. So you do a dose escalation. You'll check really low doses, you'll check high doses, and you'll figure out what is the middle point. And then, that can take a couple months. It takes longer than you think because everything takes longer than you think.
Yeah.
You want to optimize it, make sure, everything works well. Inevitably, for reasons unclear, something goes wrong with every experiment. So anticipate running everything twice. Truly, like anticipate running everything twice. Um, and then, once you have a reasonable dose, you're sure it works, then you can probably prepare your mouse experiment, assuming someone gave you something in hand that you think works. And their mouse experiment is going to take, 6 months. It'd be probably the fast, to get your mice, implant it, treat them, analyze it. But more commonly, it's like you have a pathway or you have some sort of way the cell is acting that you think is targetable. And you're going to test 10 things. And you're going to say which of these things is most promising. You're going to compare them because once you move forward, you want to move forward with your most promising thing. So if you have something that works 10%, another thing that works 50%, then 72, 78, 75, 80, you're going to maybe do some fine tuning over there to say, okay, let's run this again with all these with a little more detail. Which of these really is the best? And then, because the more you progress, the more expensive your experiments get, the longer they take. So you really want to utilize your time and funds to the best of your ability. And sometimes, measuring twice and cutting once is the best way to go. So, yeah, I would say if someone gave you a compound in hand that you think is going to work, it would probably take a year in the laboratory to demonstrate in cell lines and in mice that it's safe and effective against cancer before you even consider a trial.
And then, we should probably do another podcast on this completely about clinical trials just in general. But like, what does that look like in terms of like, okay, I'd love this to be in a clinical trial. Like how does that work? Do you work with the drug company? Do you like
It all
how
comes
does
down
that
to funding at
work?
that point.
Okay.
So sometimes you can get it funded through other organizations. Hopefully, this is something that a drug company has a financial interest in. But we've had many examples of a drug that worked really well. We thought it would be great in a trial, and then there's no company that is pursuing that drug. Maybe it's too small
Oh,
in their market
interesting.
share to take the risk.
Hmm.
And actually, HER2 may be a good example of this.
Cheers.
So that, yeah, I mean, there's 2 to 4% of patients with lung cancer have HER2 mutant disease.
Yeah.
So luckily, I think for you and the HER2 community, we actually do have some really good therapies out there, and they actually have pursued them. But that
Mm-hmm.
was a financial risk for these companies, like a big one,
Yeah.
to spend these millions and millions and millions of dollars into a small fraction of lung cancer.
Yep.
And then if they're outcompeted by the next company, they lose that, you
Right.
know.
Right.
Whereas if you have a bigger market share, then it may be more financially prudent for that company to take the risk.
Yeah.
If it
That's
was,
why I say the only minor blessing in my HER2 mutation is that HER2 is present in breast cancer and they get a hell of a lot more funding. And so
yeah,
the hope for me is any drug that is in that space, could it carry over? Not all circumstances, but the drug I'm on now did.
yeah.
So there's hope there for me at least.
No, I think there's truth to that. And it's really tough for people who have rare things
Yeah. Yeah.
because no one's gonna, it's not financially incentivized to do the trials. And even preclinically, I'll say when I say preclinically, I mean in the laboratory. If I have a really good idea for a disease biology that's in 1% of lung cancers, then the person funding that research is like, why would I fund something that could help 1% of people when I could fund something that could help 10% of people?
Mm-hmm.
And that's a good
Mm-hmm.
question. Like, really,
It is.
it's a health
Yeah.
equity issue,
Yeah.
because there's not enough funding for all the things.
Yeah.
And this is where the patient advocacy groups have really done an outstanding job of coming together, particularly when you have something rare, to say, let's get all of us together, let's do our fundraising, find the experts, and then give them the funding to do it. There's interest in the academic community, like we want to do this stuff, and that makes it really hard, when you think something's going to be really promising, but no one's going to fund it because it's not in enough people. Um, and then if you have a really promising drug and that's the only one around for that group, someone's probably going to pick that up. Like if it's 0.1% of lung cancer patients, but you're the only drug on the market, that can still be a slam dunk. Yeah. So no, it's there's a lot of complications to how things get selected and proceed forward for sure.
Well, you've helped me immensely to understand at a high level, like all the ins and outs of research. I'm sure we could do multiple podcasts on this, but this was a really good start to kind of understand that world and how it works.
Yeah, I hope I've provided some clarity. There's still so many elements to this. We didn't get a chance to get into what is basic science versus translational research versus clinical research. There's so many elements to when people think about research, what are they thinking about? That's one question I want to pose back to you is when you hear cancer research, what comes to mind?
Someone in a lab coat with a looking at looking at things under a microscope and having mice.
Okay, yeah, that's pretty much what it is. Yeah, but some people think about trials, and those are so different.
Yeah.
Like those are so different, and it's tough because I think a lot of people who are newly diagnosed with cancer and they haven't been in the oncosphere for very long, the stuff that's clinical, like looking at symptoms, understanding why does this group of people tend to get this and these people don't get this, there's low barrier of intellectual entry. Like that stuff makes sense. Like you could come up with a good amount of clinical research questions, like being a patient and knowing what you have now, like you could come up with as good of clinical research questions as I could, for sure. But once you get to the basic science, the stuff looking at receptor biology, where the new advancements are made, I think a lot of people who are new to the oncosphere, the new patient, they don't know a lot of that stuff yet because the barrier to entry is higher. Like you needed some degree of science background to know that stuff. So I think that stuff kind of gets swept under the rug a little bit, which I'm a little sensitive to because that's where my work is. So that's mostly what I'm doing is the stuff in the laboratory under the microscope. And it's interesting. People think it's very exciting and it's slow. It's slow. It's monotonous. You have to have a good idea, and as I mentioned, you can have a good idea in hand, and it'll take you a year to figure it out.
Yeah.
So it's that's one big push for this podcast is I think the people who are really doing the research, the technicians who are maybe out of undergrad and they're looking for experience, or this is what they want to do with their life is do physical experiments. The PhD researchers, these are people who don't get a chance to see patients. So they're going in there every day, changing the media on the cells, adding new drug day in, day out, analyzing their data for weeks and weeks and weeks, doing the measurements on every individual mice, every individual mouse every week, twice a week, and writing all their data down. It's very, very monotonous, high attention to detail, a lot of writing down data just to see how it's going to end up weeks or months from now. And I think one thing that's really helpful for them is to have a strong connection with the patient who can help be the anchor point for why are we doing this? Like, how does this work translate into someone I care about? And it's easy to care about the mission, but it's really hard to stay motivated when things are hard if it's not anchored in someone you know. So I think this is something where the patient community, we really all need to be working together on this stuff, and it's getting better for sure. That there is a push for that to occur more often, but I feel like they feel a bit nervous about the cancer research community and getting involved in ways because it's a bunch of nerdy people and there's this perception that they're super smart. And they're dedicated. They know a lot about that topic, but like they're not different humans than anyone else. So yeah, I think whatever opportunities people have to learn more about the labs that are in their community working on the stuff that they have, like, get involved in some way, talk to them. It'll help inspire the people who are doing the garbage pail job of going in every day and getting it done day in, day out, staring at computer screens for 10 hours a day, you know.
Yeah, I've sponsored quite a few research grants, I think that's what they're called. And it excites me that I can have my name associated with some of these projects. It gives me purpose too. So I think it works both ways.
I mean, I would love to see a world where we have our laboratory meetings, and in that laboratory meeting, when you're discussing your project on HER2, you have patients who have HER2 there who are understanding what you're doing, and asking questions, and then they're able to be that anchor point for all of us, and we just work together on this stuff. And
Yep.
people ask their questions because fundamentally, as an academic oncologist, I work at a public university. I'm paid for through public funds. I work for the public. And I think there is this big barrier between what we're doing in the laboratory and what the public sees. And that's not consistent with the mission of how my job is, I think.
Right.
Like, I think we need to be more transparent about this. So hopefully we're able to do that. It's tough because when you show your hand to say you're telling other people what you're working on, the competition kicks up. They're going to say, oh, someone else is working on this. We're working on this. Let's get our stuff out. So it's vulnerable to do that. That's a reality of research. We all deal with it.
Yeah.
People want to work together, but you don't want to lose all the investment you had. And frankly, if someone's just starting something that you're nearly done with, it's probably a kind thing to stop them from wasting their time and money. Because there's no like public repository of what everyone's doing. Like that doesn't exist. There's
It
no,
should be.
there should be.
Yeah.
And then if you want to work on that thing, you collaborate. That doesn't
Yeah.
exist. So
Yeah.
right now you have an idea, you just go try it. It costs your money, your time. And sometimes you find out, because you didn't fully interrogate the data that's published, someone maybe already did this. Or,
No,
you
that
know,
would be the worst.
that's definitely happened where, you start noticing something, oh, that's interesting. And then you deviate a little, oh, that's interesting. And then someone already found that.
Oh
And
gosh.
yeah, so, but there's no public domain about this person works on this. If you want to work on this, you work together. That's not a thing. Although that would be a better use of resources, in my opinion, and maybe a better use of equity to get more cancer researchers working actually working in total because you have less wasted funds on repetitive data. All right, I feel like we're getting close to time here.
Yeah.
What
I filled a lot.
what last question? Do you have any other last questions for me before we have to kick off?
No, I was looking at my list and we pretty much covered everything that I had without me even looking at the list that much. So I'm proud of myself.
Good job. You've thought about this a lot. I mean, your life depends on these things,
Yeah, very much
in a
so.
real way, and we have to do better, and we can do better. And I think the community is getting better at how we use cancer research funding, but there's a lot of systematic barriers to things being perfect.
Yeah.
They're big systems. There's a lot of people involved, and there's not enough funding. So
Yeah.
it'll get better over time, but we have work to do.
Well, thank you for doing what you do.
Thank you for
Appreciate it
engaging.
greatly.
Yeah. All right. Well, I think this is a great place to cut it. Thanks, everyone. Hopefully you learned something today. And we have more cancer research topics for sure, because this is an endless topic that we can talk about ad nauseum. But for now, we're going to cut it, and we will catch you next time.
Thanks, everyone.
All right. Bye. Thank you for joining us on this episode of Cracking Cancer. If you found this helpful, please like, subscribe, leave us a review, and visit crackingcancer.org for more information. Every dollar we earn goes directly to furthering the podcast and supporting vital cancer research. Because together, we can crack cancer.